All right, genetics. This is our next unit. Genetics, of course, is the study of traits and inheritance and how things are passed on from one to another, from parent to offspring. You with me? Genetics, yeah. And we're going to look at genetics from a sort of a non-biological point of view, first of all, because what you have to understand is the person who, Gregor Mendel was his name, the person who sort of is considered to be the father of genetics, the one who put it all together, did all this back in the 1800s before we knew anything about genes or DNA. He had no idea about those things. So at first, it was a concept that he put together, and then later, as we discovered these parts of cells and DNA and how it all works, we realized that these were the mechanisms by which his genetic theory actually worked. So we confirmed his ideas with actual biology later, which is kind of cool. But before we dive into that, we're going to review a couple of important processes. And what we're going to basically talk about is reproduction, cell reproduction, because in order to pass on your genetics, it has something to do, of course, with reproducing. And there's basically one way that cells reproduce themselves, and then there's another sort of variation of that when we get into sexual reproduction. So the first type of cell reproduction is called mitosis, and it's sometimes just referred to as cell division. Okay. Now, what we should understand is that cell division in mitosis is for eukaryotic cells. You remember what that means? Cells with nuclei. Bacteria cells also divide, but their division is slightly different. It's kind of the same thing, but obviously because they don't have all the different organelles and things inside, the process is somewhat different. So for bacteria, we call that binary fission. So I'm just going to put in brackets here. Mitosis occurs in eukaryotic cells. Unfortunately, when we take high school biology, we focus everything on eukaryotes, and we don't really mention the poor bacteria much. And so we get away from them. But they're quite interesting, eukaryotic cells. Bacteria also split, right? Prokaryotes. Split in a similar process, I'll put. It's kind of similar. A similar process called binary fission. Just so you know, and sort of in your head. When you get to university, you'll start to study microbiology as one of your options, possibly, if you go into bio. And when you do that, you'll get to know bacteria pretty well. So this is sort of an aside, just to remember that binary fission is also a splitting process similar to mitosis, but mitosis itself is eukaryotic cells, and these are the ones we're going to focus on. Primarily because in grade 11, our focus is human. Our focus is understanding human biology. And so we, of course, are eukaryotic, right? Except for our red blood cells, which don't have a nucleus, because they are so highly specialized for gas exchange, they've given that up, remember? Cool. Okay, so let's go through mitosis as the cell division process. So essentially, what a cell has is it has sort of a life cycle that we've divided up into phases. Biologists love to label and divide things up into groups, right? So I'm going to draw for you a simple cell cycle. And it's a circle, so it really doesn't have a particular starting point, but I'm going to do a green circle over here like this. Stop there. And then I'm going to do the blue part across the top like this. And then another green part over here like this. And then a red part down here like this. There. There's my circle. We're going to call this the cell cycle. That's what it's called, the cell cycle. And it's kind of a generalization. You have to remember, whenever we do things in biology like this, they're generalizations, and all cells go through some sort of cycle very similar to this, but it's possible that, there could be some variations. So, cell cycle. All right, let me get a smaller, let me label these things. The green part over here is called the G1 phase. The G stands for growth. So this is a phase of growth. It's where the cell is growing, and it's, doing whatever it is that it likes to do. Maybe it's a liver cell, so it's learning how to detoxify things, and it's doing cell stuff. It's just living and growing. Up here we have the S phase, the blue part. Now the S phase is a very special phase because what happens here is this is when the cell is starting to think ahead. It's starting to think about reproducing itself. And so in order to do that, the first thing it has to do is it has to make a copy of its genetic instructions. And you know that those are held in the chromosomes. And so in this phase, the cell copies chromosomes as sort of a getting ready chromosomes. Then we have another little period of time or phase where the chromosomes are copied, but there's a little more growth going on, the G2, or the second growth phase, which is again growing and doing things. And then the red part is the actual dividing. The red is the cell division or the mitosis process. So what we're going to do now is we're just going to divide this red line up into the 4 stages of mitosis. Okay? So now from now on, we're just looking at this piece of the circle to see what happens there. But we're going to remember this piece because that's kind of an important part of the process. Okay. And I'm sure you saw that in grade 10, right? Yeah, good. Some say no. If you didn't, well, you've seen it now. All right. So mitosis. Now, I'm going to talk a little bit about chromosomes first, because what happens is it becomes a little bit confusing. So let's just draw some pictures of chromosomes. And let's talk about what they are, first of all, before we go into the actual stages of mitosis. So a chromosome is made of a compound or a chemical called DNA, right? So they're made of DNA, which is a shortcut for deoxyribonucleic acid. Now, I'm going to get you a picture, an internet picture. Okay, I found a picture here of DNA. Now, this is a chemical molecular model. And what I want you to understand is that, we draw these lines. We draw squiggly lines and go, oh, that's DNA. Well, remember, everything is made of atoms. So this is going to zoom in to the DNA molecule and look at the actual atoms that are found in it. And it's actually not that complicated. It's what you call a polymer, which means it's got a little section that just keeps repeating itself over and over and over. And so if we look at this, we can see these little shapes. Now remember, in organic chemistry, there's a shortcut. Every corner where lines touch is a carbon atom. Okay? So if you look at all these little boxes, let me get some color here. Let me get red and go back here. No, don't copy this. Just to look at. If you look at all these little corners where those are all carbon atoms where I'm putting these little red dots. And then any atoms that are not carbon, we label them. So you can see nitrogen atoms and oxygen atoms. Essentially, though, this is just a mixture of carbon, hydrogen, oxygen, and nitrogen. Those are the only 4 atoms that you need to make all your genes, all your information. Yes? Okay? That's all you need to make an entire genetic instruction book that can contain 100,000 or more genes for a human being. You just need 4 types of atoms, which is pretty cool. So here's what I want to show you. What I want to show you are the components of DNA. You see this little part right here? That's a sugar. It's a kind of molecule called a sugar. If it's all by itself, we call it, there's 2 kinds. There's ribose and there's deoxyribose. Deoxyribose means right here on this carbon, there's an oxygen that's not there. That's the only difference. So that's called deoxyribose. Now over here, there's a phosphate. That's called a phosphate. You can see the P in the middle, and you'll remember from grade 10, phosphate was PO4. Remember the ion? And there's the 4 O's all around it, PO4. So that's why it's called phosphate. And then over here, we have this other guy, this big thing right here. Now this one is a little bit different. This is what we call a nitrogenous base. And the reason we call it nitrogenous is because it contains nitrogen. That's all. And we call it a base because it belongs to that big category of chemicals. Remember acids and bases? So we call it that. But there's basically 4 of them that occur. There's actually 5 that we use in genetics and in cells. There's adenine, which we commonly just call A. There's cytosine, which we commonly just call C. There's guanine, which we just call G. You know how it got its name? We found guanine, and we isolated it in bat droppings, which are commonly called guano. So we called it guanine, bat poop. That's why we named it. Then there's 2 more. There's thymine, T, right? And then there's another one called uracil. Now here's the funny thing. It's a U. There's 2 genetic molecules. There's DNA, deoxyribonucleic acid, that doesn't have an oxygen in this little spot right here. And then there's something called RNA, which is ribonucleic acid, which does have an oxygen in that spot. That's really the only difference. There's one other major difference, actually. The other major difference is that DNA contains these 4, but RNA, wherever there's a thymine, instead it has a uracil. It's a slightly different combination. So when you're talking about RNA, you'll have A, C, G, and U. But if you're talking about DNA, you'll have A, C, G, and T. But the U and the T sort of do the same job anyway. So it's just a weird quirk of nature. And that's kind of the main idea. We're going to be talking mostly about DNA. RNA comes into the picture next year when you learn about the complexities of how genes express themselves as proteins and the whole process of biochemistry there. Now, if you look at the other side here, right down the middle, whoops, I'm going to have to go back. I don't want the help desk. Sorry. If you look right here, I'm going to draw this line. You see these little dash lines that are crossing here? That's a sign of a weaker kind of bond that's holding one side of the DNA to the other. But if you look at the other side of the DNA, and I'm going to change colors to make it easier. I'll go to green here. If you look at the other side, it's really just a copy of the same thing. Here's the phosphate. Here's the ribose sugar. Actually, forget that one. This is the phosphate. That one's part of the next piece. And then here is the nitrogenous base. Right? Well, actually, yeah. Actually, it's these 3. These are the 3 that form the repeat. This one is actually part of the one that's coming up next, so actually ignore that one. Look at those 3. You can see that that's kind of an upside-down version of what's on the other side, isn't it? And so what happens is in DNA is this whole section gets repeated. If I go down here, I can see the whole section again. A phosphate, a sugar, and this time I have a different base. I have the guanine. See, this one says thymine, and this one says guanine. So the only thing that changes is you can have any one of these 4 A, C, G's, or T's attached there, right? But it's the same thing. And if I went down to the next one, it would be the same and the same and the same, except that the base might be different. Over here on the other side, I'll go back to my red color. On this side, it's the same thing, right? I have a phosphate and a sugar and a base, just like I had up here, repeated. Except that I went from adenine and now I have cytosine, slightly different base. And that's how DNA basically looks on a chemical structure. in a chemical way. It's all these little atoms all joined together. But we're going to draw simpler versions of this because this is too hard to repeat all the time. So what we're going to do is we're going to look at a simpler version here. Let me just go back on my, let me get rid of this. Here's a sort of a model of it done with little models for all the different atoms. You can see them all there. But look at this picture. This is a little simpler, right? We can see there's the sugar, there's the phosphate, and there's the base. Sugar, phosphate, base. Sugar, phosphate, base. All the way down, and it continues. And then on the other side, the same thing, sort of upside down. Sugar, phosphate, base. Sugar, phosphate, base. Now let's go to this picture. Oh, here's another one that shows several of them all sort of joined together. But let's go to, I have to erase my drawing. Let's go to this picture over here. All right. This is kind of a simplified picture where they're just using the shapes rather than the actual atoms, right? We've got the phosphate, the sugar, and then the base, A, T, T, G, C. And of course, the information of a gene is coded in the order of the bases. A, T, T, G, C is a different code than G, T, G, C, or something different. And so the order of these bases along the molecule here, that's how the information is stored in kind of like a code, a 4-letter code. It's actually a little more complicated. What happens is 3 of the bases make sort of a genetic word, and then it reads them 3 at a time. So the order makes a difference. But now look at this picture. Oh, I got to go get my pointer. Actually, you know what I can do? Use the computer to point and my finger to draw. So here, if I erase, got to erase anyway, don't I? All right. Let's erase this drawing here. Here's a picture that's kind of showing a more simplified version. So what they're showing here in this picture is red, ink, back we go. If you look right over here, the sugar phosphate forms like a backbone. It goes down like that. So now they're just drawing it as a gray line. So you see that squiggly gray line that's winding down here. That's supposed to represent the sugar phosphate that's attached like that. So it's a simpler way to draw it. And then inside they have these little colored bars. And you'll see that each bar represents a particular amino acid. So the little bars across are these, right? Like that. And they're joined. There's 2 different ones joined together on either side, one on one side, one on the other. And you can see how there's a red and a yellow. So that's adenine. This one is adenine. And on the other side, there's a thymine. So this is typically how we draw DNA when we're just doing a quick drawing. We want to draw it. We draw it as 2 squiggles, right? Like this. And we put lines across. And we say that's DNA. But now you understand that those lines represent more complicated molecules made of atoms. That's all I really wanted you to understand. Because if you just draw this, you're like, well, what the hell is that? It's a red line on a board. What is it? You know what I mean? Now we can see the greater structure. But from now on, we're probably going to use a simpler picture like this because we're concerned with the overall process and not so much the actual biochemistry. When you get to grade 12, you're going to have to learn these more complicated versions. Okay, so that's kind of a little introduction to what DNA looks like. So let's go back to our note here. Okay, and so basically we could say, all right, let's draw this in like this. And this is the phosphate and sugar backbone. Backbone, right? And then in between, we have the bases, which we can draw them. Maybe I'll draw them as different colors, but I'm going to use blue, blue. I'm putting them spaced out because I want to do something kind of cool here. And then on the other one, I'm going to put maybe a couple green ones, like that. Then I'll put some red ones, sort of in between. I should be making them a little bit longer, but that's okay. It doesn't really matter. And then on the other side, oh, I need one more color. I wonder if this yellow will show up. Yeah, it kind of does. Put the yellow sort of anywhere. The order doesn't matter for the picture. It matters greatly for your genes, but we're just drawing a picture. So I need to extend these so they're more like halfway down. That was kind of silly. Now here's the cool thing. I'm going to tell you this. I'm going to write it over here. Wherever there's an A, on the other side, there has to be a T. That's the only way they stick together. And whenever there's a C, on the other side, there has to be a G. So the two sides of the DNA molecule can only stick together in this way. So let's say that we call our red one an A, and our blue one is a T. That means across from every A, there has to be a T. So across from every red A, there's a blue T. That also means that across from every blue T, there has to be a red A. So every blue one has to have a red one attached, right? Like that. And then, if we call C the yellow one, the C always has to attach to G, which is the green one, which means across from every yellow, there's a green. Like that. And across from every green, there is a yellow. Like that. So that's how our DNA would look. And then we can just label this the nitrogenous bases. Nitrogenous bases. And we should write their names down so we remember their names. Adenine, thymine, they go together. And then the other pair is cytosine and guanine. We want to remember at least those. I can't get my page up. Guanine. At least those. Okay? And of course, we can put a little arrow here. Those are what's inside. So they form like the rungs of the ladder. So DNA is often described as a ladder with a sugar backbone, sugar and phosphate sides, and the rungs are the bases. And if you grab this ladder and imagine it's rubber, you twist it, and you make it into like a spiral. And that's kind of what the DNA molecule looks like. That's why we draw it in this weird shape. We don't draw it as a straight ladder. It's a ladder that has like a wiggly twist in it. And that's what this was supposed to look like, a twisted up ladder. Okay? Now, that's the DNA molecule. That's where the genetic information is. But remember, genetic information is simply the order of A's, T's, G's, and C's. A typical gene is a region of DNA that has many of these letters. So there could be 200 of them. So a gene would be like a whole segment of DNA like this that had, you know, it would be AAGT, TTGGG, CCGG, HGG, HCG, DTTGG, ACG, DTGG, GGG, ACT, or something like that. And there's an interesting process as to how that particular code of letters is actually the instructions for how to build a very particular protein molecule. And that's how the information is stored. Okay? So when we talk about a gene, so we're going to be talking about genes a lot, a gene is a sequence of bases, a sequence, that says sequence, it doesn't look like it, but it does, a sequence of bases along a DNA molecule that code for a specific protein molecule. Every protein in our body that our body makes has a code, and all those codes are contained in the DNA as orders of letters for a specific protein molecule. So it's like an instruction book on how to build proteins. Now what's cool is you might say, well, how do you get fats then, and how do you get other things? How come your genes just make proteins? Well, the reason is because proteins serve as the catalysts. Remember what that word means? A catalyst is something that helps a reaction happen. Proteins are the catalysts that control the building of sugars and fats and all the other molecules. So if you want to build a body, all you have to do is have instructions for all the proteins. And when you set them all loose, they cause all the other reactions to happen that builds everything else. So our genes are instructions for proteins, and the proteins regulate and control all the other processes that cause the buildup or breakdown of everything that we need to build human beings and other living things and have them live and do their thing. So it's pretty cool that it's actually fairly simple. Right? Being alive, in some ways, is fairly simple. What's complicated is how the genes actually do that and how they build the protein and all of what we call the biochemistry of what goes on underneath. Okay? So that's what we mean by a gene. It's a segment of DNA. Now, the DNA in eukaryotic cells is sort of specially packaged. If you're a bacteria in a prokaryote, let's put prokaryote here, prokaryote, right, like a bacteria cell. Inside the bacteria cell, the DNA is basically just kind of floating around in there. And it's often found in a ring structure, right? So it's all the DNA sort of all wound up in a ring. And sometimes they'll have other little rings of DNA, little smaller versions. And it just floats all around. But in a eukaryotic cell, DNA is more carefully packaged and protected and managed. The first major difference, of course, is there's an entire organelle that holds onto it, the nucleus. So in a eukaryotic cell, we have a nucleus. Okay? Let me know if I'm going too fast here. So, well, no, let's go underneath because there's too much to write. So let's go underneath here and go to eukaryotic cell. In a eukaryotic cell, which is our kinds of cells, those are cells that have a nucleus, right? So obviously, the DNA is found in the nucleus or is kept in the nucleus. It can't leave the nucleus, in fact. That's one of the problems, or one of the things that has to be overcome. Nucleus. Oops, that's kind of an... Yeah, that's an A, the nucleus. There's an organelle that protects it. But there's more protection and more organization than even that. Instead of just floating around, okay, the DNA is sort of bound all together and attached to protein molecules that are called histones. And so what happens in a eukaryote is the DNA is bound together by proteins called histones. So we're not going to draw crazy pictures, but you can imagine that you have these big molecules of protein, which are called histones, and then the DNA is sort of wrapped around like this and sort of all bound up into this structure. And this structure is called a chromosome. So the chromosome contains the DNA, but it's a little bit more complicated than just the string of DNA. It's wrapped up in proteins, and these proteins are called histones. Now the histones can stretch and contract, and the molecule can sort of change its shape, right? Because of the way it's built, it has some flexibility. And so what we normally find, in growth phases, DNA is stretched out and very thin. So you could imagine that, the histones are stretched out like this, and the DNA is winding in between. And it makes a very thin line. So thin that if you look at a microscope, you wouldn't be able to see it. And if you've ever looked at cells under microscopes, you can't see DNA normally. But something weird happens when the cell is about to split and divide itself. When the cell is about to split, during, right, or I should say, or in preparation, because it happens beforehand, in preparation for mitosis, the splitting process, the chromosomes do two things. They replicate, they copy themselves, and become short and thick. They shrink up. And when they become short and thick, we can actually see them in a microscope. Okay. So what would that look like? Well, when they replicate themselves, you get two copies of DNA. But first I'm going to draw sort of what happens. The histones all bunch up like this, right? Pulling the DNA together, and the DNA is still all wound around there. But now see how it's shorter and thicker. DNA is very long. Believe it or not, there's many meters of DNA in every one of your cells. It's hard to imagine how something so big could fit into a microscopic nucleus of a cell, but it's because it's so thin. But when they crunch all up, they get thicker. And then they double. And so you end up with a double chromosome. So then you end up with another one that's a direct copy of this one, also bunched up beside it. And they're actually stuck together. If I were to draw them, sort of, they would be stuck together sort of in this middle region. So there'd be like a piece like this hanging out the back and another piece like that stuck together. And the DNA is all wound around inside their histones. And that's what happens once they're copied and they become thick. So when we look at the microscope, we can see them. And this is what they look like. They look like little sort of X-shaped things joined in the middle. And we have learned that we can stain them with various stains, biological stains. And what happens is parts of them become banded. And you start to see these patterns of thick and thin black areas or lines on the DNA. What's interesting, though, is the patterns are the same on both, because remember, they're direct copies of each other. So you have to kind of imagine whatever the pattern is on one side, it's the same on the other. And then they're joined in the middle by this structure called a centromere. Thanks, sir. A centromere is sort of the little thing in the center that sticks the two copies together. So when we describe chromosomes, we often draw them like this, and you'll see pictures like this. But you have to understand, this is not a simple chromosome. It's two copies of a chromosome that have joined together. And that's only what happens when cells divide. So that's actually two chromosomes stuck together. And when the two of them, we have to have names for them. So we still call this entire thing a chromosome, even though it's actually two of them. And then we call each piece a chromatid. A chromatid is sort of, it used to be a chromosome when it was by itself, but then it copied itself and stuck to its copy, and now it's called a chromatid, and then two chromatids make up the new copied chromosome. And so there's confusion, right? Because, and this is why meiosis is confusing. Some of you said that you found it confusing. It's because we're not thinking of the chromosomes as doubled copies when we look at them like this. And that's an important thing to remember. This is a doubled copied chromosome in the eukaryotic cell. So a lot of the times they exist like this, and we don't see them, and they're just doing their thing, and they're replicating or they're making proteins. But when it comes time to divide, they double up and shorten and get thick like this, and they stick together like this. So this is the picture of a doubled chromosome ready to divide. Got it? Good. Now we can talk about mitosis. So, let's go. Now remember, when we talk about mitosis, I'll just go back, we're talking about the bottom of that cell cycle, right there. We're just talking about what happens between here and here from now on, okay? So, the first part of mitosis is called prophase. Pro means first or before, so we have prophase. That's the first stage. So here's our cell in prophase. So the first thing that happens is the nucleus begins to fall apart. Because in order for our chromosomes to sort of be spread apart, we can't have a nucleus in the way. The nucleus traps them. So the nuclear membrane starts degrading, starts falling apart. So the nuclear, whoops, I don't want that guy. The nuclear membrane dissociates, falls apart, or disintegrates. Let's put disintegrates. Now remember, it's just a membrane that's made up of little fat molecules. Remember the model of the membrane? So they all just kind of fall apart and float around, and the membrane disappears. Later, that membrane can reconstitute itself when those molecules can come back together. So it's kind of cool. And the chromosomes inside are already copied, because remember, they copy in the S phase, which happens way long ago in the cell cycle. So inside here, we have copied chromosomes, which I'm going to draw in different colors. I'm going to put 4. Here's a little copied green one. Here's a little copied blue one. Here's a little copied red one. And here's a little copied purple one. Now, different cells have different numbers of chromosomes, right? Humans, we have 46, whereas other animals might have 42, or some things have hundreds of chromosomes, 112 or something. And it's weird, because the things that have more chromosomes are sometimes more simple organisms, like wheat. I think wheat cells have over 100 chromosomes. But a human, which seems much more complicated, only has 42. So it's kind of interesting. But we're only going to draw 4 because it's too hard to keep track of 42 of them, right? The picture would get too crazy. But all 42 in a human do the same thing. Okay? Now it turns out that our genes are interesting. We actually have sort of, they say a double copy, but not quite. So I want you to think about this example. Let's say you had 2 firefighters. They're both the same job, right? So when a fire comes out, both of those firefighters run to the fire and put it out. But interestingly enough, one of the firefighters is a man and one is a woman. And they both have slightly different methods of fighting the fire, but they accomplish the same task. So we would say that those two are not exactly the same, but they're so close to the same job, right? We call that homologous. That's a homologue. Somebody who does the same job as you, so they're slightly different in their appearance and how they do the job, but they accomplish the same task. 2 firefighters. In our cells, in humans, there are 46 chromosomes, but they are always in pairs of homologues, which means there are 2 firefighters, 2 dentists, 2 lawyers, 2 of everything. So our chromosomes can be grouped as 23 homologous pairs, right? So here, we're going to pretend that the blue and the purple are homologues. And we're going to pretend that the green and the red are homologues. So what that means is that even though they're different chromosomes, they both do the same job, which means they both have the very same kinds of genes. It's just that the genes might be a little different. So for instance, on the blue chromosome, there's a gene for controlling the production of melanin, which is a dark brown pigment. On the purple one, there's also a gene for controlling the production of melanin, a dark brown pigment. But they don't have to be the same. The blue one could produce more melanin than the purple one. So both do the same job. They both make melanin, but one's a little different. One makes more than the other. So if a person gets the blue one, which makes a little more, then they will have darker eyes, or darker hair, or darker skin. But the person who gets the purple one will have lighter skin or lighter hair. That's the difference between homologs. They do the same thing, but they do it slightly differently. That's not important right now for mitosis, but it's very important later on in genetics and in meiosis when we get to that point. So just kind of remember, these are homologs, and what that means, right? That they perform the same job. So they can be grouped like this. But for mitosis, it doesn't really matter yet. I just wanted to bring that up so that when we talk about it again later, it makes sense. Okay, so that's what's happening in prophase. The chromosomes are already copied. The nuclear membrane is beginning to disintegrate. The other thing that's happening is little structures called centrioles. Actually, let me draw them somewhere else. They're floating around in the cell. And what they're doing is they're moving to the ends of the cell. They're starting to find their way to the two ends. And you'll see why in a minute. So those are called the centrioles. Centrioles, that yellow is hard to see. And they're moving to the other ends of the cell. That's prophase. Good? Can we move on? Okay. Now we got to get to metaphase, which is the next stage. Metaphase. In metaphase, the whole cell is just sort of beginning to elongate, but not much. And what's happening is the centrioles have moved to the ends. The chromosomes have all started to line up in the middle, like this. What's my other color? Green? No. Purple? Red? Yeah, green. So there they are. They line up in the middle in metaphase. And the other thing that happens is these tiny structures called spindle fibers. They're like little ropes. They begin to form. And they attach to the centriole. Let me use black for this. And they start to reach out like this. And they begin to attach. Now what they do is they attach right where the centromere is, in the middle. Like that. That one's going to attach later because he's in the wrong sort of spot. And so when they attach, they can pull, and what they can do is sort of rip apart these two copies that were joined together, remember? That's their role. We call that metaphase. So spindle fibers are forming. You can write that beside her. Spindle fibers are forming, and the chromosomes have lined up along the center of the cell. Those are the distinguishing features here. Then we have anaphase. In anaphase, the cell begins to stretch. So we have an elongated cell. It's definitely stretching a lot. And as it stretches, the centrioles are pulling on the spindle fibers, which are causing the little copies, so red first, the little copies are being yanked by the middle. So they tend to look like little V's because they're being pulled by the middle, like this. And the purple guy. And the green one. They're being pulled by the spindle fibers. Pulled like this towards the two ends of the cell. And the cell is stretching. So now what we've done is we've pulled apart the two little copies. See that? And then we move to the last phase, which is telophase. And in telophase, we have the cell now. I'm going to move down a bit because I need more room. I'll go back up if you want to finish the picture later. The cell then begins to take on this hourglass shape. The spindle fibers have done their job. They have pulled a copy of each of the chromosomes onto one side of the cell, like this. Purple. Then what happens is a nucleus begins to reform. Oops, black, black, black. Our nucleus begins to reform on both sides to contain the... So we could write over here, the nucleus reforms, and the cytoplasm and the membrane pinches off in the middle. Eventually, this middle part will squeeze together and touch. And when it touches, the membranes will just snap apart, hang on, and we'll end up with two complete new cells. And so now we have two complete cells. Now what's interesting is they both have exactly the same chromosomes that the first cell had. If we go back to the first cell, we'll see that it had purple, red, blue, and green. The only difference is they were doubled up here, so they were in their doubled copied form. Before they were copied, they would have just been single lines. And so when we go down here, we see that we now have the same thing, a purple, a blue, a green, and a red in each cell. So we've made a genetic copy that's supposedly identical. Now that doesn't mean it's always identical, because in the process, sometimes weird things can happen. Sometimes anaphase messes up, and a chromosome might get stuck together and not be pulled apart. And extra chromosomes would go to one side and not the other. And it would create two new cells with very different chromosomes. And whether or not they lived or died would depend on which chromosomes moved and how important they were. And sometimes they would live and just sort of be different. Other times they would die. So mistakes can happen. Mistakes can happen in the process, but typically they don't happen, and this is what we get. So that's mitosis, and because we're out of time, we'll stop there. And then we'll talk about how this is used for some kinds of reproduction.